Good morning, everybody. Thank you for joining us today. My name is Rhonda Henshaw-Powell, and I'll be your moderator for today. We have an exciting presentation for you. And without much further ado, I'd like to hand over to the host of today's event, Dr. Phatak Farahani. Thank you.
Thank you. Hello, everyone. My name is Phatak Farahani, Marketing Manager at Advanced Cell Diagnostics. I'm your host for today. Welcome to ACD's Knowledge Resource webinar series, where we'll give you closer understanding of how RNAscope technology is contributing to specific application areas. We begin our knowledge series today on a topic of stem cell research presented by Jyoti Phatak-Sheldon. Jyoti holds a master's degree in molecular cell biology along with bachelor's in dental surgery. She has previously worked in the field of genomics and precision diagnostics as a product specialist for qPCR-based liquid biopsy tests. Jyoti currently works at ACD as an associate scientist in the application team to execute projects and collaborations that demonstrate novel applications of RNAscope, BaseScope, and microRNAscope assays. We'll now begin.
Hello everyone, and thank you so much for joining the Knowledge Resource Series webinar for stem cell research. My name is Jyoti Sheldon, and I work for the applications team at ACD. We'll be reviewing today how you could spatially map genetic variants at a single cell resolution using the RNAscope technology for your stem cell research. So first, let's understand what is the RNAscope technology. It is an in situ hybridization assay, which is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in both cells and tissues, giving you that morphological context at a single cell level. Now, the technology consists of three parts. It consists of the unique target probe, which ACD will design against your gene of interest. It also consists of this signal amplification system that generates a high signal-to-noise ratio. And lastly, your data can be visualized as single RNA molecule or dots. This assay allows you for spatial mapping of various types of RNAs, such as mRNAs, link RNAs, splice variants, highly homologous sequences, also short sequences, and point mutations. And you can do this in both cells as well as intact tissues. Data can be visualized either as a fluorescent or with a chromogenic label. You can perform this assay on a wide variety of sample types. The sample types does depend on what assay you're using, but most of our assays work with FFPE tissues, fresh and fixed frozen tissues, and we also have protocols for PBMCs and cultured cells. So now let's get a little bit of understanding of how you can achieve that high sensitivity and specificity using this assay. The two key features of the RNAscope technology are its probe design and its signal amplification. This oligonucleotide target-specific probe is depicted as a Z to emphasize the fact that they have two regions that are linked by this spacer. Now, the bottom of the Z complements and hybridizes to your gene of interest or the target transcript, while this top of the Z serves as a base for the amplification structure that will be built. When both these Z's hybridize in tandem to the target sequence, it creates this binding site upon which a preamplifier can bind and the amplification tree can be built. Now, after the Z pairs hybridize with the target RNA, the preamplifier binds to the top of the Z pair. And to this preamplifier, you can bind multiple amplifiers. And to each of these amplifiers, you can bind multiple label probes. This label probe contains either a chromogenic or a fluorophore that generates a signal detectable under a standard bright field microscope or a fluorescent microscope. Now, this particular signal amplification strategy yields high sensitivity and allows you to visualize your target RNA as a single dot, where each of these dots will represent an individual RNA molecule that can be further quantified. Background is eliminated because of the signal is dependent on both of these Z's binding next to each other on the target sequence. What that means is if you don't have both of these Z's sit right next to each other, this tree won't get built, and therefore you won't have any signal. Consequently, no amplification of non-specific hybridization will occur, generating little to no background signal. Now, a standard RNAscope probe will consist of about 20 Z pairs pulled together that are designed to hybridize right next to each other on your sequence of interest. However, you only need 3 of these Z pairs to bind to get you enough amount of amplification and signal potential. And this is really important when you're working with archival FFPE samples that can be degraded. And so this technique really allows you to get that high sensitivity in those kinds of sample types. So taken together, the combination of a probe design and signal amplification ensures you a high signal to noise ratio. So under this RNAscope technology, ACD offers 3 unique assays for spatial mapping of gene expression. The first is the standard RNAscope assay. The standard RNAscope assay consists of about 20 Z pairs, as I mentioned earlier, per target. You can use it for detection of both mRNAs or long non-coding RNA targets that can be anywhere over 300 nucleotides. This is available as a singleplex or a duplex assay or a multiplex assay where you can go to up to 12 targets simultaneously on a single tissue section. And the singleplex, duplex, and the multiplex 4-plex assay can be performed either on an automated platform such as Leica or Ventana or a chromogenic platform. The second offering under this technology is the BaseScope assay. The BaseScope assay can consist either a 1 Z pair or up to 3 Z pairs depending on the target of your interest. It can be used to detect splice variants, short, highly homologous sequences, and point mutations. So targets anywhere in the range from 50 to 300 nucleotides. It is also available as a singleplex chromogenic assay or a duplex chromogenic assay. And then we have our newest assay, which is the microRNAscope assay. Now this assay allows you for detection of ASOs, microRNAs, siRNAs, and smaller sequences anywhere between 70 to 50 nucleotides. Currently, this assay is available as a singleplex chromogenic assay, which can be performed either on an automated or a manual platform. This is the broad product portfolio that fall under the RNAscope technology. As I talked about earlier, you have the RNAscope chromogenic, which can be used as either as a red or brown or a duplex assay. You have the multiplex fluorescent that allows you to look up to 4 targets simultaneously. Or you have the new RNAscope HiPlex that allows up to 12 targets simultaneously. You also have the BaseScope singleplex or duplex assays, and you have the microRNAscope assays that allow for short targets. We are coming up with an expansion of the RNAscope HiPlex assay very soon that will allow you to detect up to 48 targets simultaneously on a single tissue section. If you want to learn more about this assay, stay tuned for our new product introduction webinar and you can get more details on it. In addition to all of these targets, ACD offers a wide variety of catalog probes of over 26,000 probes currently available. Although, if your probe of choice is not available in catalog, and as long as you know the sequence of your gene of interest, probes can be designed for any targets from any species in as little as 2 weeks. So with all of that product portfolio, let's dive deeper into how RNAscope can be applied in various areas of stem cell research. Applications include detection of molecules such as GPCRs, including Lgr5. Lgr5, as you know, is a target that's known to have challenging antibody for protein detection by IHC, as well as long non-coding RNA, which cannot be detected by IHC since they do not code for a protein. In addition, one can visualize differentiation of induced pluripotent stem cells, or iPSCs, as well as organoids, to characterize novel cell types in the tissue context. CRISPR-mediated gene editing at the transcript level, as well as splice variants, can also be visualized with our BaseScope assay that I talked about earlier. And lastly, with that ushering era of single-cell technology, the multiplexing capabilities of the RNAscope assay allows for both confirmation as well as spatial mapping of gene signatures identified through transcriptomic approaches such as single-cell RNA sequencing. So now I've picked a couple of publications that show you some of these applications applied in the field, as well as some in-house data for applications in stem cell research. As we talked about earlier, Lgr5, an epithelial stem cell marker in GPCR, has been well recognized as difficult to specifically detect by IHC in both mouse as well as human tissues. Therefore, an alternative is to use RNAscope to detect this gene in the tissue context. To date, there have been over 50 publications for detection of Lgr5 using the RNAscope technology, and this number just keeps on increasing with time. So this is some in-house data where we wanted to visualize the spatial orientation of the Lgr5 positive cell population within the intestinal epithelium. We used the RNAscope probes designed against known markers of several cell populations in the small intestine. Enterocytes were detected with intestinal alkaline phosphatase, goblet cells were detected with mucin 2, enteroendocrine cells were detected with chromogranin A, we detected PNAS cells with lysozyme 1, and finally, the columnar-based crypt cells, where the intestinal stem cell population resides, were visualized with either Lgr5 and olfactometin 4, really showing you the importance for the role of RNAscope in giving you the morphological context in your data to be able to visualize where exactly a gene is being expressed in a tissue context. Additionally, we visualize the intestinal stem cell population that resides in the base of the crypt using the RNAscope multiplex fluorescent assays. What you see are known markers again of the intestinal stem cells, with Lgr5 being in white, ASCL2 in green, and olfactometin 4 in red. So this really shows or highlights the importance of RNAscope in studying co-localization. As you see, these three signals co-localize in cells that are located primarily at the base of the intestinal crypt, really highlighting the application of this technology to detect your target of choice and then locate them in a morphological space using various known cell markers. So we also examined internally the localization of receptors and ligands involved in the Wnt signaling pathway in intestine. As you know, the Wnt signaling pathway plays a very crucial role in stem cell biology. In this image, we show the spatial expression patterns of the ligand Wnt3A and the receptor respondin-3 in both the non-inflamed colon as well as the inflamed region. Interestingly, we saw that expression of respondin-3 was detected primarily in the submucosa and the muscularis mucosa, while Lgr5 was primarily detected in the neighboring mucosa of the base of the crypt. Another application of RNAscope that is used commonly, not just in stem cell but all their field, is being able to compare the tissue context of normal tissue versus diseased tissue, or in this case, an inflamed tissue, something that really gives power to studying the morphological context in your research. So let's look at this particular publication from a customer. The hair follicle cycle goes through various stages of rest, growth, and destruction throughout life. These stages are known as telogen, anagen, and catagen. Now, hair follicle stem cells, or HFSCs, they reside in the outer bulge of the follicle and are the regenerative source of cycling hair follicles. But how they maintain their stemness is poorly understood. So in this particular paper from Institute of Medical Biology in Singapore, Lim and other researchers determined that the autocrine Wnt signaling in outer bulge cells maintains stem cell potency throughout telogen and anagen, while paracrine Wnt inhibition of inner bulge cells reinforces differentiation. They used the RNAscope chromogenic red assay to demonstrate varied expression levels of multiple Wnt's in the bulge cells during telogen and anagen cycles. This is another paper where iPSCs, or induced pluripotent stem cells, were known to become an important tool for modeling and investigating human diseases as well as for screening drugs. So really important in drug therapies or screening of drugs. Now, following differentiation, thorough molecular characterization of the cellular identity is critical to understand this. Now, RNAscope technology is well suited for confirming this cellular identity of iPSC-derived cells. For example, in this publication, researchers at Janssen, they evaluated and thoroughly characterized commercially available human iPSC-derived neurons. RNAscope was used to confirm neuronal and GABAergic identity of the iPSC-derived cultured neurons, demonstrating that these human iPSC-derived neurons were primarily expressing both MAP2 and VGAT. As you know, VGAT is a GABAergic marker. This is another example of a publication from a customer. Now, intestinal epithelial cell renewal is controlled by regulatory signals produced by the epithelium itself as well as the surrounding cells. Although there is evidence that stromal cells contribute to the intestinal stem cell niche, their markers and the soluble signals they produced have been completely undefined. So in this particular paper from Amsterdam University Medical Center, Olga Karpis and fellow scientists show that CD90 positive fibroblasts are located in close proximity to stem cells in vivo and support organoid growth in vitro and produce class 3 semaphorins. Now, they used dual-ish IF, where you can simultaneously visualize both your RNA target using RNAscope, as well as an antibody using this dual-ish IF sequential workflow to visualize CD90 positive fibroblast localization in the mouse script cells, thereby revealing that the CD90 is a marker of colonic stem cell niche fibroblasts. In addition, they also performed the same sequential RNA-ish IF workflow and identified the cellular source of class 3 semaphorins, or the soluble mediators involved in signaling from fibroblasts to stem cells. This dual-ish IF workflow has been widely applied with our RNAscope customers and users because it really gives you the power to visualize both RNA as well as protein on the same tissue context, reducing your number of experiments. The previous sequential workflow that is used by this customer works in a way where you performed RNAscope first, and then you perform the antibody detection to look at the simultaneous RNA protein on the same tissue context. We did recently come up with a new version of this workflow called the co-detection workflow, and I will talk about it in some of the later slides. So now I wanted to kind of focus on how RNAscope has been used for organoid research. Organoids are another important tool for modeling and investigating human diseases and screening for drugs, and their use in the field has gone up significantly in the last few years. The RNAscope technology has been widely used on a variety of organoid tissues. For example, the assay has been used to show the human endometrial organoids recapitulate their molecular signature of the glands observed in vivo. In addition, it has been used as a pancreatic tumor organoid library to identify functional subtypes based on their stem cell niche factor dependencies on Wnt and respondin. And lastly, it has been used to discern species-specific detection of genes, such as Lgr5 in human intestinal organoids that were transplanted into mice. So I'm going to take a couple of examples of these papers and go more in detail. This particular paper is from University of Cambridge, and they define a method of establishing genetically stable endometrial organoids that can be cultured long-term and recapitulate the gene expression patterns of endometrial glands in vivo and functional characteristics of their cell of origin. So in this paper, they used RNAscope-ish to compare the molecular signatures of endometrial gland markers between both the endometrium tissue as well as the organoids, really once again highlighting the use or application of RNAscope, giving you that morphological context and the tissue context. And these methods will be an invaluable research tool for researchers to study newer therapies for common pathologies of the endometrium. This is another paper. Now, transcriptomic studies have shown a strong correlation between human intestinal organoids, HIOs, and fetal tissue compared to adults and developed mature architecture after transplantation. Now, this paper is from University of Michigan Medical School, Ann Arbor. Now, they used RNAscope red assay to compare Lgr5 expression levels between these HIOs, or human intestinal organoids. These HIOs were transplanted in kidney capsule of immunocompromised NSG mice and adult human intestinal tissues. What this paper is suggesting is that these HIOs represent the fetal immature intestine, and they also identified several hallmarks of fetal to adult intestinal development that can be further modeled in HIOs that can be transplanted into mice. So with these examples of organoids, I'd now like to move into single cell research. As I talked about before, single cell research has really just kind of shot up in the last couple of years, giving the importance of studying single cell transcriptomics in the field. We're seeing more and more customers performing single cell analyses to reveal the heterogeneity in complex tissue organs. So initiatives such as these transcriptomic cell atlases, they require assessing gene expression at that single cell level. However, the current single cell transcriptomic techniques utilize dissociated cells, and this results in the loss of spatial organization of the cell population in that tissue context. So validation and spatial mapping of single cell analysis can be obtained using assays that retain this spatial organization, such as the RNAscope in situ hybridization assay. With its robust, specific, and sensitive methodology, along with its multiplexing capabilities, you can confirm and validate and spatially map your high-throughput single-cell transcriptomic results. Now, this technology will allow you for cell type specific expression profiles to be mapped back into those complex tissue contexts of these heterogeneous organs. Some of the applications that we've seen in single cell research are to spatially map these transcriptomic cell atlases, to visualize gene signatures of some of these newly identified cell types, and I will show you an example of this in a project later on. You can classify highly heterogeneous cell types such as the brain or the tumor microenvironment. You can confirm new therapeutic cell types. Also, you can characterize immune landscapes in response to diseases or infections. You can analyze or predict responses to drug treatments. And you can also confirm some of the publicly available data sets, examples such as the TCGA or the tabular mirrors. So with some of these examples, I'd like to show you how customers have applied RNAscope in single cell research in the field of stem cells. Now, the murine epidermis and its hair follicles is an ideal model system for tissue regeneration and stem cell research. However, it is a surprisingly highly heterogeneous tissue. We're seeing this hair follicle is a really sought after field in stem cell research. So in this particular publication, researchers from the Karolinska Institute, they performed a single cell RNA sequencing to identify 1,422 single cell transcriptomes and 25 distinct populations of interfollicular and follicular epidermal cells. They further then used the RNAscope assay to validate their results with the correct spatial organization and reveal heterogeneity among the stem cell compartments. So also in this paper, these authors took the sequential dual RNAscope-ish IF workflow and validated their RNAseq data and mapped these defined cell populations to their spatial location in the epidermis. Again, highlighting the role of morphological validation and confirmation of your single cell data in stem cell research. So this is another paper on stem cell research. During homeostasis, Lgr5 positive CRIP cells drive turnover of the intestinal epithelium. However, even when these Lgr5 positive cells are lost during injury, the epithelium somehow still recovers. So therefore, there had to be an additional population of quiescent stem cells that exist to drive epithelial renewal in response to injury. So researchers from Alex Gregorius' lab at Mount Sinai Hospital in Toronto used single-cell RNA sequencing to profile the regenerating mouse intestine and identify a novel stem cell population termed as RevSCs or revival stem cells. And you can see them here in the cluster SSC2. What they found were that these stem cells were enriched for genes involved in cell survival, such as the clusterin or CLU. Now, during homeostasis, these CLU-expressing cells were rare, and they were distinct from these Lgr5 positive cells. But they also saw that in response to irradiation, the number of these clue-positive cell epithelial cells expands, so basically in response to injury. Now, they used the RNAscope multiplex fluorescent assay and revealed that these rare clue-positive cells in the normal intestinal crypt were distinct from these Lgr5 positive cells, as you can see in the image on the left. And they saw that there was a significant increase in these clue-positive cells in the damaged intestinal crypt. Furthermore, the researchers identified YAP1, a transcriptional regulator, and is important for regeneration of this intestinal epithelium. So during intestinal regeneration after injury, so after a response to an injury, the YAP1 induced the reversal stem cells that will create new Lgr5 positive cells and thereby restore the epithelium. Single-cell RNA sequencing revealed that this YAP1 gene signature was present exclusively in the SITU cluster that we saw earlier, and that deletion of YAP1 in the intestinal epithelium suppressed the emergence of these endogenous clue-positive cells that we were talking about. And this was demonstrated by, again, using our sequential dual RNAscope ISH and IF workflow. So we talked a lot about this dual RNAscope ISH IF workflow, and I mentioned to you earlier that the previous workflow went where you would first perform your RNAscope assay, followed by performing your detection for your antibody. But we recently launched our new co-detection workflow, which allows you, enables researchers to rescue previously incompatible antibodies that would not work with our sequential workflow when you combine it with ISH. It also reduces the need for an extensive antibody clone screening for co-detection. If you want to learn more about this workflow, we will have our new product introduction webinar, or you can also contact our support team and get more information on this. So what are some of the applications other than the ones we've seen for this co-detection? You can correlate, of course, your RNA protein expression. You can examine cell type specific gene expression. You can also validate newer antibodies, and I've seen a lot in this in the field where there isn't a tissue-based method that currently exists to validate antibodies. So this serves as a great tool for that, and we're seeing more and more customers use it for this application. You can visualize cellular localization of RNA targets. You can also identify cellular source of secreted proteins. You can also discriminate antibody detection of multiple targets, including splice variants and highly homologous targets. And you can detect pathogens in different replication states with your host cell markers. So with all of this, I'd like to introduce and give you a little bit more information on our RNAscope HiPlex assay. As I mentioned earlier, the RNAscope HiPlex assay allows for visualization of up to 12 mRNA targets on the same tissue section. So how does this assay work? It requires an iterative fluorescent detection of 3 different rounds with 4 targets each. So 4 multiplied by 3, you get 12 targets. Between each target, there is a cleaving step, but this cleaving procedure is rapid and has minimal to no effect on your RNA and tissue morphology. Now, precise image registration algorithm, or our HiPlex image registration algorithm, what it does is at the end of these 3 rounds, it allows you to register all your 3 images together and give you a true visualization of your data at that single cell level. Currently, this assay is only compatible with our fresh frozen or our fixed frozen sample types. But we have in our pipeline, it isn't works that this assay will be compatible with our FFPE cell type down the road. So to show the application of this assay in our single cell field, we use an example of a paper. This is a paper by Goksha published in Cell Reports in 2016, where researchers at Stanford identified novel subtypes of the D1 and the D2 striatal medium spiny neurons by analyzing over 1,200 striatal cells using single-cell RNA sequencing. They identified discrete cell types that exist in a continuous spectrum of transcriptional states and found that functional diversity within a complex tissue arises from a small number of discrete cell types. However, they did not confirm their single-cell RNA sequencing results, nor did they show the spatial relationship between these D1 and D2 subtypes. Therefore, we employed the RNAscope technology to confirm and spatially map these novel cell types in the mouse brain. We did this by performing the RNAscope HiPlex assay in its two different formats. The first is the RNAscope HiPlex 8 reagent format, where we looked at the D1 and the D2 subtypes individually. And then we also performed them as the RNAscope HiPlex 12 format, where we looked at all the targets on the same tissue section. So this is a still image of the RNAscope HiPlex 12 reagent, where we looked at both the D1 and D2 population. So you saw that image earlier, and there's so much of that data going on. So one may ask, how do I really understand when there's so many targets together? And for that, I wanted to highlight the use of our HiPlex image registration software. Now, those researchers from Stanford highlighted an important finding in which they identified a group of cells that express both the D1 as well as the D2 genes. So using our HiPlex image registration software, you can toggle on and off your genes of interest as you see on the right. The image on the right is the same frame as you saw in the previous image with all the 12 targets. But using our registration software, I only selected for the genes of my interest and identified that novel population that expressed both the D1 and the D2 subtypes, really highlighting not just the use of our HiPlex 12 assay or the HiPlex kit, but also this image registration software in allowing you to resolve this complex heterogeneous tissue data. So with all of the single cell research, I did want to move on to a topic that is not related to stem cell research, but given the current pandemic situation and knowing how all labs have sort of diverted their focus into understanding the nitty-gritty of this COVID-19, I did want to move into understanding how RNAscope can be applied in your COVID-19 research. So the RNAscope product offerings can be applied across a wide range for COVID-19 viral detection, starting with understanding or detecting the virus itself using our singleplex assays. Using our duplex assays, you can look or understand at the virus receptor interaction. We know some of the common receptors that were used in the field early during this research were ACE2 and TIMPRESS, so we have probes available for those already. As a lot of you may know, the COVID-19 infection is followed up by this cytokine storm. So using our multiplex assays, you can look at the virus receptor interaction, but you can also look at this immune response that follows up. And in addition, we've seen a lot of the field, people in the field look at some histopathological markers post-infection. So using our assays, you can also look at that. But most importantly, with our HiPlex assay, you can look at this entire plethora of probes and understand the entire pathogenesis of how this virus is acting through its infection and working in the body. And so with this morphological context, the specificity, and the single molecule detection of RNAscope, you can look at that entire mechanism of action of the COVID-19 virus. In addition to the probes, standalone probes, based on some early literature, we saw some commonly used probes by the customers in the field. And so we pulled together some of these targets and bundled them as these COVID-19 probe sets that are now available for customers to buy, where some of these targets are in pre-assigned channels to detect and study the viral pathogenesis in human FFPE tissues. You have the ability to plex up to 4 targets and save some of these precious samples that are hard to get for studying your COVID-19 research. So how is RNAscope applied by our customers? Currently in the COVID-19 literature out there, Based on some early literature and the new literature, we've seen that the 4 main topics that RNAscope has been applied is for studying cell and tissue tropism, for studying natural immunity, for vaccine development, as well as for placental transmission. So let's go over some of these just briefly. To understand natural immunity, the questions that were being asked in the field were, can natural immunity be induced by infection, right? So once a person is infected, is there an immune system that happens where it can prevent the person from being reinfected? So some of these papers that you see out here used RNAscope to visualize tissues and cells that were infected by SARS-CoV-2 and then further demonstrate viral replication. So we have probes that allow you to look at not just the viral strand, but as well as the replicating strand of the virus, really highlighting the importance of RNAscope in this research for studying natural immunity or immune response in animal models. So as you see here, RNAscope was used to characterize the lung pathology by demonstrated by multiple clustered site of infections and by high infection in the ciliated bronchial epithelial cells. And our SENSE probe was further demonstrated an active viral replication in the lungs. The next area that RNAscope was used is because of its ultrasensitivity. Customers used it in vaccine evaluation. The 2 papers that were used, where RNAscope was used for Moderna mRNA 1273 vaccine evaluation, as well as the thermostable AR-CoV vaccine. And the questions really were, how well are these vaccines working, right? We do know there's over 160 vaccines that are currently in development, but are these vaccines really working? So RNAscope was used to answer some of these questions where customers visualized the viral infection in control animals and then confirmed the absence of virus in these tissues in vaccine-treated animals. So sort of like a before vaccine and after vaccine tissue analysis. This is an example where RNAscope was used to study placental transmission. So this particular study that you're seeing is from Luisa Patani and other researchers, where they studied 2 cases that were SARS-CoV-2 PCR positive for mother, neonate, and placenta. They used our RNAscope 2.5 Leica Brown assay to visualize the virus in the placental tissue and evaluate the distribution or the tissue distribution while retaining the morphological context. RNAscope validated the presence of the SARS-CoV-2 RNA in the infected placentas, supporting the possibility of a vertical transmission of SARS-CoV-2 from mother to baby in utero. While I showed you a couple of examples, we do have a wide range of publications that were used amongst these various application areas for COVID-19 research. Now, if you'd like to access this list, you can reach out to our support team or anybody at ACD, and we can provide you this information, as well as you can visit our webpage, which is dedicated for COVID-19, and get more information on the probes, as well as some of these publications as well. So talking of all of these publications that I showed you for stem cell as well as COVID-19, I did want to highlight that the growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then, there have been over 2,700 peer-reviewed papers published using the RNAscope technology in numerous journals, and many of them in top-tier ones such as Nature and Cell. Now, the RNAscope technology is highly relevant across multiple fields of research. Our top areas are cancer, neuroscience, and infectious. But we've seen a lot of applications in areas such as stem cells, as we talked about, development, inflammation, and such. But we are seeing more and more application of RNAscope in single-cell RNA sequencing workflows, demonstrating that rapid growth of publication using this assay to confirm and spatially map single-cell RNA sequencing data in tissue context. So with all of these publications and with all of this data, I hope I've convinced you and to also summarize today that the RNAscope uses a unique probe design strategy to dramatically improve your signal-to-noise ratio. We currently have over 22,000 catalog probes, but like I mentioned before, if you know your gene of interest, we can design new probes for any target and any species in about 2 weeks. The probe design and the signal amplification strategy yields in a high sensitivity and specificity of this assay, really allowing you to work with even low expressing targets, degraded FFPE samples, and some complicated sample types. Our multiplexing assays, along with our HiPlex assays and the new expansion that will come out soon, will allow you to detect up to 48 targets simultaneously in the single cell and tissue context. But with that being said, you can also plex down. If you don't need that many, we have options with our 4-plex assays and 12-plex assay as well. Our other assays such as SpaceScope and microRNAscope assay will allow you to detect targets such as splice variants and short sequences down to approximately 50 nucleotides and 17 nucleotides. And lastly, our dual-ish IHC workflow, both the old sequential as well as the new co-detection workflow, allows you to simultaneously detect RNA and protein on the same slide, really giving a lot of power to your data and giving you this whole package of your data analysis. So with all of that, I conclude my presentation today. Thank you so much for attending today's webinar. I hope you enjoyed it, and I hope you're all staying safe. If you have any questions at all on the technology, on our applications, or you need any information, you can contact me on the email on the screen. You can also reach out to our support team for any technical questions, or you can also visit our website and get some information on that. Thank you again. Thanks for joining.